Hey guys, welcome to chapter 17.1. All of chapter 17 is about gene expression, and 17.1 specifically is going to be kind of an overview of the central dogma. And so at this point, I'm sure you know that the central dogma is DNA, RNA, protein, and that means that our information on how to make proteins starts in our DNA. It gets transcribed into RNA, and then it gets translated into proteins. And so gene expression is the process by which DNA directs the synthesis of proteins. And essentially, our proteins, their job is to express our phenotype. And Archibald Garrod was the first to suggest that genes dictate phenotypes through enzymes. He did this um, by studying a disease. He said that diseases can be caused by enzymes being made improperly or enzymes not functioning. And he looked at a disease called alkaptonuria, which is a disease where an enzyme is being made incorrectly. And the enzyme cannot break down the chemical L-captin. And L-captin, when it builds up in your system and it's exposed to air, it turns brown. And so people with this disease or that can't make this enzyme are going to produce brown urine, or they're going to have brown pigment in the whites of their eyes or in their ears. And so he said that when if the enzyme isn't made, then there's going to be a disease that follows because the enzyme has a function and it's not able to function. Later work was done by Beadle and Tatum. They worked on bread mold. So what they did is they grew bread mold spores on a petri dish. And a medium is the agar in which it grows, and there's a food supply in that agar. They provided only a minimal nutrient medium to the bread spores. And the minimal nutrient medium is the food that they give them has only very small molecules in them. And so the food has to be built up, or what the bread spores have to do is they have to take those small pieces and make bigger pieces. So they take the small pieces or the small molecules and build them up into large molecules like amino acids. And then with those amino acids, they can make proteins. Proteins are very important to life, and so you have to have all of your amino acids. And so this was the wild type of bread spore. They said that that the wild type of bread spore that lives in nature, they are able to take small pieces of, they were to take small molecules and be able to build them up. And then what they did is they generated nutritional mutants. They exposed them to colonies, they exposed the colonies to X-rays. And those colonies could no longer synthesize, take the small pieces and build them up into larger pieces like amino acids. And so when they grew those bread spore on a petri dish with minimal nutrient medium, then they were unable to survive because they didn't have the enzymes to build those amino acids. And what they did is they created a complete medium where those mutants were able to grow because the food in that agar contained entire amino acids. So those small pieces were already put together. And therefore, they could take those amino acids out of the dish or out of the medium and use it for themselves. So they didn't have to take the small pieces and build them. And so they were able to survive because they didn't have the enzymes to build them, but they didn't need them. So they hypothesized that each nutritional mutant contained a mutated gene for a specific enzyme that had been disabled. So they created minimal medium, but then also put valine, which is an amino acid. So it was all the small pieces and then one type of big piece. And then another minimal medium, but lysine. Lysine is another amino acid, so it wouldn't have to build lysine. And then another minimal medium plus arginine. And in the vials grew specific molds that were able only to grow on arginine because they did not have the enzyme to produce arginine. So they were able to grow on this medium. The conclusion was that this spore was missing the enzyme to synthesize arginine. And this resulted in the one gene, one enzyme hypothesis. This hypothesis is going to change as time goes on, but it started out as the one gene, one enzyme hypothesis. So because it couldn't make the arginine, it could grow on the arginine medium because it lacked the enzyme to make arginine. So another two colleagues used a collection of arginine requiring mutants to investigate a biological pathway for the synthesis of arginine. And by using tests with different compounds and different numbers of compounds, they realized that arginine, in order to make arginine, it took 3 different compounds, which means that the metabolic pathway in order to generate or to synthesize arginine has 3 different enzymes. And so the conclusion, there are 3 steps in the metabolic pathway to synthesize arginine. So as scientists continued their experiments, they learned that not all proteins were enzymes. So for example, the hormone insulin is a protein, but it is not an enzyme. And the structural protein for hair, keratin, not enzymes. So it changed to the one gene, one protein hypothesis. Then scientists realized that many proteins are composed of more than just one polypeptide. For example, if we look at hemoglobin, it has 4 polypeptides. And so therefore they changed the hypothesis to one gene, one polypeptide hypothesis. Now this one gene, one polypeptide hypothesis is not entirely correct, but it's a good overview. But the 2 reasons that it's not a good, entirely correct is because, one, a eukaryotic gene can potentially code for a set of closely related polypeptides, meaning that in order to express a trait, you might have a gene can contain related or multiple polypeptides, and they can be used in some polypeptides can be used in other proteins, and so it's just not so simple. So we call this process alternative splicing. So a eukaryotic gene can potentially code for many related polypeptides, and they don't all code for just the same protein all the time. So there are a number of genes also that code for RNA, and that RNA is not all going to be translated into proteins. Some they just do jobs in throughout the cell, and so it doesn't really ring true as one gene, one polypeptide. But it is a good overview. So let's talk about the central dogma. We talked about the experiments. Now let's talk about the big idea of central dogma. And this is how proteins are made. This is how we express our genes. And it is DNA, RNA, proteins. This is coined by Francis Crick. And this little arrow right here between DNA and RNA, this is going to be, this represents transcription. And transcription is the process of transcribing information from DNA, here we go, to RNA. Here it is. So this is transcription right here. And translation is the synthesis of a polypeptide using information from mRNA. So so translation is represented by this arrow. We are translating the information from A's, T's, C's, and G's, the genetic code, into our amino acids, into the language of amino acids. When we are talking about transcription or DNA replication, anytime we're talking about a polymerase, DNA polymerase or RNA polymerase, remember that we use read up and write down. So when we are reading our DNA, we are reading up from 3' to 5'. But we are going to write down. We're going to write from the 5' direction to the 3' direction. Read up, write down. And I want you to remember read up, write down. And then when we're talking about translation, of course, it's biology, switch that. But don't try and remember the words in the opposite order. Just know that when it's a ribosome doing the job, it's opposite. But it's polymerase, it's read up, write down, and the ribosome just does it backwards. So remember that when we go through and it starts talking about 5', 3', and it gets confusing, just go back to that. Polymerase reads up and writes down. Ribosomes do it backwards. And so the principles of transcription and translation. We learn about ribosomes being organelles. However, they're not technically an organelle. They're not membrane-bound organelles. then they exist also in bacteria cells, which means they're pretty ancient. So in a bacteria cell, the DNA is going to exist in the cytoplasm. And so therefore, all of the DNA, the central dogma is going to occur in the cytoplasm as well. And so the second that this the second that the RNA polymerase is transcribed, it can immediately go into translation because the ribosome can hang out right near that DNA. And so it can produce proteins quicker than eukaryotic cells because they're able to all exist together in that cytoplasm. Eukaryotic cells are going to be a little bit different. Translation occurs in the nucleus. So here is our eukaryotic cell. And here's our gigantic oversized nucleus in a eukaryotic cell. And notice here that we have an extra step. So here's our DNA. And the polymerase is going to, RNA polymerase 2 is going to read that DNA. It's going to transcribe a pre-mRNA strand. And then that pre-mRNA strand is going to go through something called RNA processing, which we'll talk about in 17.3, I think. And after RNA processing, we get this strand of mRNA called the primary transcript. It is the primary transcript that actually leaves the nucleus. And then the primary transcript then goes to the ribosome, which is in the cytoplasm. And there is where it gets translated into amino acids, or into the language of amino acids, or into a protein. When scientists realized that DNA was the transforming molecule, or DNA was the molecule of heredity, they weren't sure about how many nucleotides it took to represent or to code for a single amino acid. And they said it doesn't make sense for it to be one nucleotide to one amino acid, because then we could only have 4 amino acids, right? So if the T represented an amino acid, well then we would only have an amino acid for T, G, C, and A. So then they said, well, it could be 2 nucleotides that code for an amino acid. And that didn't make sense either, because we knew that there's 20 amino acids. And this could only make 16. So that would be like if T and G represented an amino acid, or if G and A represented amino acid. But that didn't work. So they came up with the triplet code. Remember that the triplet code exists on the DNA. And a codon exists on the mRNA, or on RNA. So the triplet code is that is the 3 nucleotides on the DNA that code for an amino acid. And so here we see that the RNA, so the triplet code codes for a codon. The codon is on the RNA, and then the codon codes for an amino acid. So triplet code, codon, amino acid. When we're looking at the DNA, a gene is going to be read by DNA polymerase. And so if the gene is on this bottom side, this is going to be the template strand that it's reading. And it's going to always be on the template strand. It's always going to read it from the same side. That gene's going to always be on the same side. However, not all genes are on the same side. Another gene could be on the other side. But if that second gene is on the other side, it will always be on that second side. So now that we have transcribed, we've talked about transcription and DNA or RNA polymerase transcribing into an mRNA strand, creating that primary transcript. When that primary transcript leaves the nucleus and goes to a ribosome, it's going to be read, but it's going to be read, like we said, opposite, not read up, write down, opposite, the other way. And so the ribosome is going to read the codon, and it's going to be the 3-letter code. And if it's a UAC, then we're going to start, if this is our codon wheel here, U, and then our second one, A, and then C, and that lines up right here, and that's going to code for tyrosine. And notice that most of the amino acids like tyrosine, serine, cysteine, they have multiple codes or multiple codons that code for each of the amino acids. And it's important that we know that the direction is read in the opposite way because we don't want it to be read the wrong way. The message would be different, right? It's important that we know that the dog runs. That's different from snur god et. So that is the genetic code. And just one last tidbit, the genetic code is very nearly universal, except for slight variations in mitochondrial DNA and some archaeans. Archaeans are different and very cool. Bacteria use the same genetic code as vertebrates do. So CGC is always going to code for proline. And so this allows us to do really cool things. For example, we take a gene from a tobacco plant or from a firefly and we implant it into a tobacco plant. And so because the fireflies use the same genetic code as tobacco plants, it can still be expressed. It's still the same mechanism. And so here we have a pig expressing a jellyfish gene because the genetic code is universal. We have the same enzymes and the same polymerases doing the same jobs, reading the same genetic codes. All right, I hope that helps, and let me know if you have any questions.